Hi everyone, and welcome back to the Information Bottleneck. And today, as always, we have Ellen. Hey, Ellen.
Hi, Ravid. It's good to be here. And hi, it's Oded.
Yes, Oded.
Nice
Yes.
to meet you, Oded.
Very nice to meet you.
And yeah, so today we are going to talk about the review process and the AI and grants in AI and what we can actually and how can we use AI in order to make better processes. And for this, we have today Oded Rechavi. He's a professor at Tel Aviv University, a biologist, and also the co-founder of QED Science, the AI peer review platform. Hi, Ed.
Hi. Thanks a lot for inviting me.
And thanks for coming. So I think like we can start with maybe like the current process or like the current problems, if you want, in the review systems. So tell us, like, why you actually thought about this idea? Like, what are the problems that bring you to this field?
I mean, academia has many problems. We still love it, and there are lots of advantages to being an academic. But one thing I think that most researchers, if not all of them, or everyone in academia will agree on is that the publishing, scientific publishing, is totally messed up. It differs between different fields. I am a biologist, so I know the situation best in the life sciences, but it's true, I think, throughout, that it's just a huge pain to get your work published, to get your work also funded, and to navigate the literature and understand what is worth pursuing, what can you build on, and what can you trust. And we use the method of peer review, which is not that ancient, in fact. It exists for a few decades, and it's the best system we have, but it's pretty bad. So I started. My motivation for starting an AI company was to change this process. But when we started doing that, and we'll go into that very deeply, we understood that the problem is much bigger. And I'll elaborate about the problems with peer review. But in the end, the problem is that, at least in biology, we still don't have a good method for validating science. Perhaps it's a little bit different in computer science. Still, you need to review tons of papers. You're nodding. You're saying no, but in biology, until you know whether something actually works, it can take decades sometimes, so a long time. And it's very hard to replicate studies. It's extremely expensive and hard technically, and no one has the motivation to do it because it will be harder to publish. So we just don't know what works and what doesn't work. And this is across science. It's just a really big problem. The way that we currently deal with it is that for every paper that you submit, assuming the editor finds it interesting, and after you submit it to a journal, there are a few of your peers that go over it and try to understand whether it's worth publishing or not. And when they do that, they have to overcome all kinds of biases, conflicts of interest, and just the fact that it's extremely hard to review papers, and it's becoming harder and harder in biology for sure, but probably also in other fields.
So was there like a specific paper or rejection that actually like pushed you to this field?
And there are so many. I mean, for years, I'm quite known for making jokes about this in social media, for just making fun
Right.
of.
I just want to, I didn't say it, right? Like, Oded, like, is like famous on Twitter, right? That like he's publishing memes about like funny memes about like jokes that like related to publications and things like that. So yeah, you should follow him on Twitter.
Yeah, so I've been
Sorry,
doing
continue.
it for many. So I've been doing it for many years, making jokes about the process. But it's really, the humor is there to deal with the sad reality, which is this really hurting us. I mean, it's wasting a huge amount of time. Half of my time I spent trying to get papers published and grants accepted. So it's really the time that is left in the end for actually thinking about interesting things or for enjoying the profession is lost. It's demoralizing. The more experience you have, the less it hurts you. And if you are already tenured and promoted and so on, it's not the end of the world. There are worse things in life, but still, people, for young people, especially for students, postdoc, and so, it's a great cause of suffering. And I also think that today, when we're dealing with many people abandoning academia, moving to the industry or to do just other things, part of it is because of that. You do the research, you are excited about it, and when it's time to actually show it to the world, it can take years. And throughout these years, you are humiliated, ridiculed, and really being tortured. And this is because of, in part, scarcity of resources and just limited resources and human nature. So that's the sad reality. There are all kinds of workshops for training peer reviewers to be nicer and so on. But in the end, it's human nature. When there are limited resources and reviewers are hidden behind, they have their anonymity. We don't know who they are. There would be bad behaviors, and this is something that just happens, and no matter what you try, you can't stop it. And not only does it delay science, my personal record is 6 years between preprint, we'll explain perhaps preprint in a second, and actual publication. So 6 years, that's crazy. Normally for me, it's a year or two. But it's not only that, it's also the way science is being written. I think that people in advance, they know what they're going to face, so they allow themselves less freedom, they try to say less, so they won't, press on anyone's toes, and they also sometimes cheat or sort of, cut corners because they know that they might be picked on if they say something that doesn't fly smoothly by the reviewers. So I think it really hurts science.
But do you think so? Like, what are the actual problems here? Like, is it like the human nature? Is it like something like we just don't pay the reviewers enough? Do you think now if we'll pay, like, you will have a lot of like money and we'll pay for a lot of reviewers, like we will get better results?
So it's a very complicated question. So first of all, it is also tied to the economic reality of scientific publishing. As everyone knows, there are just a few publishers, very big companies making a lot of money, and they have pretty much a perpetuum mobile where they don't do so much. I mean, we write the papers, we pay to publish our own papers. If we're talking about the top journals, it can be even $15,000 or so to publish one paper. And then we also pay to read our own papers. The universities pay millions to subscribe to different publishers, and we also do the review work for free. And since in the end, everything depends on this publication. It's what they call the publish or perish, and your promotions and your prestige and tenure and hiring and all of that depends on that. This is the only currency. And there are not so many jobs and not so many grants, and so it becomes a competition. And in the end, the people who are most suited for reviewing your work are your competitors. Most people, I want to believe, are really good actors. They do it to improve science, and that's fine. But there will also be bad actors, and when people are pushed to the limit, when they don't have resources, they don't have time. Imagine that to review a paper or a grant, I have to dedicate the whole day. No one is paying me for that, okay? And so they might do a lousy job, and they can delay you to move ahead. They can steal your ideas. As I said, in most cases, they are good actors. They won't do that. But even if they have the best intentions, it's just extremely difficult to do with limited time. And even if you have all the time in the world. Biology papers are really long now. They contain tons of information. No one reads every supplemental figure and so on. And not to mention, go over the, repeat the experiments or redo the analysis. No one does this. I mean, maybe, a handful of people, but not many people. So it's limitations of time, ability, and also lots of built-in conflicts of interest, and the fact that the whole thing is tied up to this economic model, and so this is why it doesn't work. And I'll say that because of that, we know that the problem is only becoming worse and worse as time passes. Now most journals can't find reviewers. So maybe the top journals, Nature, Science, and Cell, can find reviewers. It's also hard for these journals, I believe. But for the smaller journals, they're just incapable of finding reviewers. So in the end, after inviting 20 reviewers, they settle for number 21. This is a person who just doesn't know what you're working on, and this is how it looks. Yeah. So that is the problem in a nutshell.
And what is why not to go to open source journals?
Yeah. So what happened was, so I'll mention also preprints in some fields, in computer science, physics, math. You had the archives that exist for a long time, and people are used to putting everything there, right? In biology, it's relatively new. I don't know exactly for how long does it exist, probably 15 years or something like that. And we have a bioarchive and med archive for medicine papers. And but still a relatively small percentage of the community post preprints. I preprint all my work, but I think the studies showed it's about 10% of papers or less, something like that. Still, and this solves some of the problem. The work is disseminated fast, it's online, everyone can read it. So that handles the distribution and also the cost problem. The thing is that you don't know whether what you're reading is actually valid. So we don't have, if it's not reviewed, so you don't know that it's, you don't have this step. And plus, it doesn't satisfy the demand for some sort of a grade for how good it is, which because committees, hiring committees, tenure committees, promotion committees, because they can't read all the papers, it's just so hard. They need some kind of heuristics to know who to promote and who to hire. And currently, they use the very bad heuristic of just the journal name. They say, okay, it was published in Nature, let's give this person a job. And that's a very bad way, but this is how it works currently. And one paper in these good journals can make or break your career.
And
so oh, sorry. Well. So with all of these publications having so much positive upside, what do you think that there's anything that the conferences or even the journals can start doing to maybe push back on this dynamic just a little bit, or do you think that peer review is almost inevitably in this position?
I think peer review, this system is, it's very hard to change. So there are all kinds of attempts in the end. They were all, none of them really moved the needle. In fact, some of the efforts might have been sort of counterproductive, perhaps. For example, because now there's more demand for opening everything for open science, and what happened was that, and this is of course a great thing. We want the information to be available for everyone, but the solution is just preprinting it. But what happens when you want to publish the paper in a journal that it will be open access? The journals move the cost to the authors, so they also just pay more to open the paper, and this also changes the financial model of the journals. Most journals were used to to selling subscriptions to universities. Now, if the papers are open, they get paid per paper. So to survive, they need to publish more papers, and then it just makes things worse because they can't review them properly, and the quality drops. And the only players who survive this are the big publishers, the for-profit publishers, and the small ones, the society journals and so on, they die or get acquired by the big ones and then lose their character. So I think that within the system, it's extremely hard. This is why we think that, the system just needs a total rethink. And we need to think about what do we want in the end? We want the information out there as fast as possible, and some sort of a badge for how valid and good it is. And we want this, and we want a transparent analysis so that you can look at it and not only get a grade in the end, which will always be a shallow, but you'll be able to dive deep and understand exactly what is strong and weak about the paper, where it needs to be improved, what is the follow-up work that needs to be done, and so on. And I think, and this is why we started QED, that this can be to some degree automated. And when you introduced in the beginning of the recording, you said, that we are building a peer review system, an AI peer review system. I don't think of our system as peer review. I think of it as review. I mean, the AI is not your peer; it's different. And I think that we don't want to mimic the current system. We want to give it a different look at it from a different angle. And so also in the design, everything that we do, we don't try to dress up like humans. We explicitly put emphasis on the differences and the parts that the machines are better than the humans.
So just 1 minute before we will go deep into what you actually offer or what you suggest, just one. So in AI, I think most of the time, the more prestigious volume is like conference, right? Like the 4 big or the 3 big conferences, and that you can submit for free, and you only pay to go, and the registration, and everyone, at least now the current form is that if you're submitting something to these conferences, you actually you must do review, and the reviewers are mostly PhD students, postdocs, or even MA students. The cycles are really short, probably 3 months from submission to the acceptance, or even less. There is almost no rebuttals, right? You basically have 1 week to do the rebuttals. And there are a lot of maybe we can talk a bit about the pros and cons of this system, and why not to go to these type of systems also in other field. And I think the pros here are you see them, right? Computer science or AI, the progress is really fast. The time scales are much shorter. You can get most of the time, you get the reviewers, again, fast, high visibility. You can have, right, these days, the citations and citation numbers of these conferences, and the age index is huge. But as we all know, as our audience probably know, it's very noisy, right? The reviewers some of them are low quality at the end, they doing poor job, and sometimes it's not, it's almost, you feel that it's quite random, that you can send and you can get some feedback, and to the next conference you get different feedback. How do you see this system? How do you see this system, review system? Do you think it can work for the long term?
So I think that certainly the system that you have is much better than ours, for sure. Right? On objective measures, just the fact that it's very fast, just a few months. For us, it's much longer. The fact that when you submit to a conference, you must review for this conference. This is also great. For us, it doesn't work like that. I don't know if the conferences that you are talking about are for profit or not, but no, so they're not. So this is something else. It's a community effort. For us, there is a system already in place, which is totally for profit, entrenched, very rich, very powerful, and it's very hard to move, to change the architecture of how it works. In the end, I mean, there are many advantages to your system. I mean, there are 0 advantages to our system. Okay, so your system is just better in every way. But in the end, also the system in your field, as you said, the job in the end is to try to see whether to do a good job reviewing, and there it fails, right? As you said. In biology, I think it's great that PhD students and postdocs will be involved in the review process, but it's hard. It's hard. Science in biology, there's a lot of accumulated knowledge that perhaps you need to know the history of the field and some perspective that perhaps professors have that not all postdocs have. And some postdocs will be much better than the professors. Also, it's just very hard to systemize it and to ensure that this is the case. And the bottom line is, in the end, it's a sort of a ceremony that is conducted in your field and in our field. A sort of let's put it through this process of review, but it doesn't work because we cannot separate good science from bad science and valid ideas from ideas that fail. So what are we doing? That's why it's there. Perhaps in biology, there's more in stake because we don't work in parallel also in Google or and so on. So I don't know. So perhaps all so we rely more on these publications than computer scientists do. You tell me. I don't know. And also the science costs a lot more. So we work on every. You said that some researchers in your field publish 50 papers a year. For me, every publication I work on it for 5 years, or more, 8 years. For a very long time, it costs millions. It's a lot of labor. It's very different. But in the end, both systems fail. Your situation is better, but both systems fail. And this is the idea. It's not about replacing peer review or not replace peer. It's about finding a good way to validate science, which is also the ultimate goal of QED. We are building this validation machine that will help scientists with their day-to-day activities, which is getting feedback on their work, papers, grants, the papers that they read, also not the ones that they author, the things that take a great amount of our time. So the science is strength, and at the end, and in parallel, we want to learn from them how to build this validation machine that will do a better review, that will be able to distinguish wrong from right, or something that works with the literature or doesn't work with the literature, or that has gaps in knowledge or missing controls and so on. We want to build this machine that can then be applied to everything science.
So tell us.
Well, I'm also just curious, do you think, because there's at least some people in the AI publishing community who argue that the journals like TMLR and JMLR are higher quality because they force people to slow down, and that you're going to go through a process that'll take 2 or 3 years of them kind of telling you what is it, like conditional accept, and then they give you like all these changes you have to make before they actually do it. And then but we're in AI, we're one of the only fields where conferences actually rank higher than journals. So do you think that either of these factors are things you've observed? How is it in biology?
So what is better?
So conferences or journals. So in first of all, in biology, conferences, presenting in conferences, it doesn't count as publications. Okay. It doesn't count it doesn't work like that in biology. It's only papers that count. Of course, it looks good on your CV that you presented in the most prestigious conferences. But this is mostly, but it doesn't count as publication. In the end, in the CV, is the publications that matter. And conferences they select also abstracts, but at least half of the speakers in most conferences or more are invited speakers who are being selected because people already know them and they're famous, or the reputation precedes them. And so it doesn't work the same. I think it's great that conferences is the thing, as it is in your field, also in this age of AI, and we can talk about that, where you don't know to what degree is the human involved in the actual research. It's good to have the human there presenting in person, talking about the work. Then you see that it's really, they're on top of their game and what they're presenting. So I think there are many advantages to going to conferences and going more and more to face-to-face type of things. In biology, that just doesn't work like that. And to the other part of your question, whether the fact that it takes a long time to publish and you're being sort of pressure tested Whether it improves the work, absolutely. I totally think it does. So to some degree, it's also a feature, not only a bug, that it takes a long time to publish and the work gets better. However, as reality shows, we see that many times the revisions that we are asked to do are totally irrelevant. The studies show that there's very little overlap between what different reviewers ask you to do, right? And sometimes feels very arbitrary. So it's far from ideal. And I think that if there was a system, this is part of what we're trying to do, that forces you to improve your science, but in a manner that fits you, because in the end, you know what directions are the most interesting and where the work can and should improve. If you get the proper feedback, then you can gain from you can get the gain without the suffering in a much faster and more direct way.
So tell us how you build such a system.
So the first thing that we do, by the way, it's moving so fast. I mean, you're in AI, it's just unbelievable how fast it moves. But we announced QED. When I mean announced, we didn't do any PR. It's just me on Twitter, pretty much. In October, and we got when we said we have an AI review system for papers. We got many thousands of people join very fast, thousands of PIs from every place around the world, every institution, and people in the end, it was most people were really happy about it. I don't know if you remember, Rabit, but the reactions were by and large very, very positive.
Mm-hmm.
But also there was also a concern about what? I mean, you're moving it, you're replacing humans. So there were three types of emotions: happiness, and the negative emotions were fear of being replaced, and also fear they won't be good enough. And you are much more aware of the limitations at the cutting edge of AI than I am, but it's clear that it's getting better all the time. And I think many of the arguments or many of the voices you used to hear about why it's not good or will never be good will never be able to replace these voices are quieted down gradually. So it's getting better and better at reviewing, and it will probably continue to be better. And then this question remains of whether we want to be replaced or whether we should be replaced, and what is the role of the humans and so on. So our approach was very clear. It's like what I said before, that we don't think of it as peer review. We think of it as a review, which is complementary to human review. And we are learning from the scientists and we are serving the scientists. So there are many companies that want to build AI scientists and replace humans. We are not one of these companies. In QED, it's about the scientist at the center helping you. We want to help you do a better job, which means that we give you feedback. We don't write for you. We give you comments on your work, in-depth comments on whatever you do, write papers or grants, and we help you improve it. And we also, another thing that we do, which differentiates us, is that the review that we give you is very structured. This is one of the features people like most about QED. So when you upload a paper or a grant to QED, it gets broken into a claim tree, which are the claims, like a blueprint of your paper or grant, just the claims or the aims of your grant or the claims of your paper, what you want to say, structured in a very intuitive way, so you can understand where are the weak parts and the strong parts of the paper. And then for every claim that you make, we find gaps or things that can be improved, and we also suggest ways to do it, either textual changes or actual experiments you can do. And then the idea is that you will improve your paper or your grant. You will repeat this process a few times. It might take, in biology, it might take time. Maybe you'll go back to the bench and do more experiments. We'll save the different versions so people can see the improvement if you choose to present it. And then in the future, we are not there yet fully, but we will go there. We will also create a sort of a report for you. So you can say, this is the process I've been through. This is how I improved my work. And then you can submit this together with your paper or just connect it to your work. We are already implemented in bioRxiv, so there's a button there to go through QED. And then it sort of saves the need for publishing it, because you already have you have the paper out there, and you have the report of how, and the process, you can see how it was improved and so on. When we do this, we also deal to some degree with problems of provenance, or was it written by AI? Was it faked? So we are not fraud detectors at all. We don't try to do that. However, so there are other companies, looking for duplicated figures, stuff like that. I think that is relatively easy, and there are lots of companies doing that. We don't do that. The way we go about it is that because we make the process of improving the paper transparent, and because we monitor the paper throughout its creation or the grant, okay? It's sort of like a birth certificate. You know when the paper was born and how it improved. So it's like a passport. You're there throughout the entire life of the paper, so it wasn't made up overnight because you've been there and you saw
but
how
the
they
idea is to have but the idea is the goal is to have that now I have an idea, I will write it down in the system, and then the system will track how this idea is evolving, and what will be the actual experiment that I'm doing along the way, or only okay, now I have kind of finish, or a finished draft of the paper, and now I'm doing the review, and it suggests me some final adjustments.
I mean, you can do both. Ideally, you start very early, and then you'll save yourself time. As I said, we find gaps in your arguments, and we suggest ways to improve it. In the beginning, we only suggested experiments, and then we also suggested these textual improvements, like tone down your claims, or if you mention this, it will be stronger, and so on. And this is the feature people love most, because if they upload a paper late in the game, just a minute before they're submitting it, or maybe after they even preprint it, then they say, I don't have the power now to go back to the bench and do 6 months of experiment. It would have been nice if you told me that a long time ago when I just started. Then you'd save me a lot of time. But in the end, the goal is for you to, when the paper is out, because it comes with this analysis, and because you improve it to your satisfaction as much as you want, you can keep improving it. It's sort of it's a live thing. Then when it's out there already, preprinted plus QED'd, then it's like it's like when you start your car and you have the different signals that tell you that you have gas there, and then nothing is broken and you can start driving. It just comes already vetted, built in. Or when you go to the airport and you do the security check in advance and you save time later, if you do this process, then you're already ready. And then the question is, how do we we hopefully improve the situation when it comes to assessing the validity of the work. And then the question is, how do we also deal with the prestige problem, the fact that people want to be ranked, they want to be and I think that we'll move more and more to reports that actually tells you which papers are better, more reliable, more original, and so on. And then you can also use this report for that, and you don't need just the name of the journals. We don't need which are very bad as a proxy. And I think if you do that, you improve parts of the system. And the same goes for grants. We didn't elaborate about grants, but that is the big problem. In biology, we need a lot of money. Every paper, the good papers, they sometimes cost millions, right? So we need tons of money all the time. There are very limited resources. All of the big funding agencies, or many of the big funding agencies, are cutting down. You probably saw this: NIH, the NSF, ERC. So the competition becomes more fierce, and you need to write better grants. And at the same time,
just say a few words about what does it mean to actually write a grant? What is the process? How much time does it take? What you actually need to do?
Yeah, so there are studies. We also did lots of interviews, and in biology, in life sciences, people spend between 100 days and 180 days a year writing grants, something like that. It's ridiculous.
PIs.
Okay, PIs, professors, PIs, exactly. So really a significant amount of our time, and 90% of the grants that we write, we don't get. It's really bad. I think, if I'm not mistaken, when it comes to the NIH, the acceptance rate is 4% or something like that of grants. For ELC, which is a big grant in Europe, I think it's 15% or 10% or something like that. So it's pretty bad. In most grants, these are the numbers. And so what we do is we spend months writing a grant with very little outside feedback. It's very hard to know whether you do something bad or not. So the system that we are building for grants sort of gives you puts a mirror in front of you, so you know how competitive you are. That's very important. Whether you also fit the call. QED for grants. And then another problem with grants is that you get the feedback from the funding agency after a very long time, most of the time 6 months or more. So by the time you will get the feedback, in many cases the feedback is very minimal. Sometimes there's no feedback, it's just rejected for this and this reason. Sometimes there is a little bit of feedback, but it's so far in the future that you don't even remember what you wrote. It's very hard to improve like that. And the funding agencies that face the same problem as journals, they don't have human reviewers to review the grants properly. Just last week, there was a big episode. I don't know if you follow this with the ERC. The ERC, these are the big grants in Europe for the European Research Council. We live and die based on these grants. This is the best grant, and this is we need to be competitive in Europe. It's really important to have an ERC grant. And what happened is that the ERC announced that because they don't have reviewers, what will happen now is that if you submit a grant and you fail, you get a B grade and you're not accepted, you will be prohibited from submitting another ERC grant for more than 3 years, which is just it's death. It's a death sentence for if you're a new PI, you just started your lab, you're dead. Okay. There were a lot of protests. We wrote an open letter. They changed their mind, which is great. They listen. It's great. Okay, but the problem remains. They have a very hard time reviewing the papers, reviewing the grants, and you don't know whether the reviewers will do a good job. So the same problem is there. And what we're doing is we're just collecting lots of feedback from the experts on what is a good comment, what is a good suggestion. Where is the AI blind and where is it good? And we are implementing this in making a system that actually helps you improve. And we constantly rely on this feedback from the community to do it.
So you think like, but like at the end, like you have the question of like, do we have like, okay, so now I apply to grant and get your feedback, but like the question if this feedback is aligned with the, I don't know, the NSF or like the ERC feedback, right? Because if it's not, so like who
Of course.
cares, right?
Absolutely. So the thing that we're doing is we are studying what works and what fails. Okay, and the comments are tuned to that.
Can you give me an example, like what types of things like you found that actually like
Sure, I'll
align
give
with
you
the truth?
I'll give you an example which from my field. Okay, so we can talk about it later. But I work on the using a model system which is a nematode, a worm. It's called C. elegans. Okay?
Maybe we talk about it later.
Yeah, we can talk about it later.
If we
Maybe
have time.
the computer scientists are saying, "What worm? Why is he studying worms?" But I'm very not special in that there are many thousands of people studying C. elegans. It's a model system that has unparalleled success really since the year 2000. I think 8 Nobel Prizes were awarded to people studying C. elegans. But in C. elegans, evidence show that you need to, this is just an example, you need to use the power of the model, which is that it's a very powerful model for doing unbiased genetic screens. So, including these in your, such a plan in your grants will be a very good idea. So this is a very specific, you need to be properly tailored for the format of the grant, for what the grant requires. You need to tune exactly the level of risk that you're taking for a grant. Okay, some of it is in the instructions of the grants, like the ERC says that it funds high risk, high gain project. But does it really? Or not so much? So in the end, it's defining what is the right amount of risk, what is the right amount of feasibility in this, how many preliminary results you really need to have. So some things are very specific about the science, and some things are very specific, are just general fit. And it is also connected to your own research. What are your strengths? What are you known for? Where is your weaknesses? And this is also about your institution. Lots of different things.
So, like the idea is that like you collect all this information from the specific PI or from the specific experiment, and then you say, okay, so like these are the factors that like we actually care about. So like based on the information on the past and like your own research, that these are the suggestions or like the lines of directions of improvement that you actually need to do.
So we find, first of all, we find gaps. There could be many gaps that you can correct. And these could be missing controls or inaccuracies, or also things where it doesn't fit with the literature, or there's a conflict, there's a problem. Okay, we find this. We also look at the specific things that you do, the equipment that you have, the type of experiments that you're used to running. And we try to tailor the suggestions to what you can actually do. And also we are we collect the context. So we suggest experiments and find gaps in alignment with the standards in your own field. We can always, and of course, the AI will always find more and more problems. It can suggest, do the same experiment on the moon. Fine, it would be amazing, but I mean, you won't do it because your field doesn't require it. So it's all of this tailoring and the dedication to the details that will in the end work. And we are also working with lots of institutions, getting feedback to do this. And the same thing. I mean, when I said that we are working we are on the author side, I also meant that we're not building, and this is in contrast to perhaps other entities, we're not building a system to help the journals. Of course, the journals can also, gain a lot. They need reviewers. They need to screen through more papers faster and cheaper and so on. But we are not creating a tool for the journals to do this. We are on the author side, but we do collaborate with the good journals, the journals that are for science, working with the community. So among our collaborators, we have eLife, EMBO, Review Commons, and others. Same with foundations. We're working with the good ones to sort of improve the system as a whole and learn from them, and also gain credibility with the community that we're doing the right thing. So, and I think this is also a big difference, because it really depends on in the hands of who you put this power in. I'm not sure I said that correctly in English yet, but yeah.
Well, so I'm curious, I think we could talk forever about peer review, and all of the problems associated with it. But you work in biology, and I hear before the call that you know a thing or two about like RNA. So I guess I'd love to slightly pivot and ask you, maybe to start with, do you have any opinions on things like personalized medicine, which I believe is like DNA and maybe RNA based? Please correct me.
Right. So I work on lots of different things in biology. Most of them are really not applied. So I do basic science in my science. So this, QED is the first like something that I did actually works in the world. The rest is really just basic science. I work with model organisms, studying things, really doing curiosity-based science. There are lots of breakthroughs in biology that are translated to medicine later on. If I try to connect it to AI, I think that probably also this will be accelerated, but perhaps the buzz is too strong, or too much is being promised. And because biology is very, very hard. I think that where we can use AI is really to gain additional confidence and validity in what we already know and don't know, before we can just connect the dots and accelerate science dramatically, like some companies promise.
We can talk about biology. Like, this is the nice thing that you have your own podcast. We can talk about whatever we want. So like, let's talk about
Sure.
biology. Let's tell us why, like, what you're doing, and like,
Sure.
why you think it's the most interesting thing in the world.
Okay, so I do lots of things in biology. I'll tell you a few. Okay, the nice things about biology, and why I think it's the best thing in the world, as you said, is that really we are always dealing with the unknown, and it's limitless. People are talking about solving biology, not going to happen. Okay, it's really endless. You cannot solve it. There's so many variables, so many unknowns, so many experiments that just need to be done every day so we can progress. I work not only, but mostly using a nematode, a worm that is called C. elegans, which is sort of like the hydrogen atom of biology. It's a very simple, relatively simple animal that is that for this reason is very easy to understand, relatively easy to understand, but still very hard. And you can learn a lot about it. So this worm has just 959 cells, out of which 302 are neurons. So a third of its body is a brain. It's transparent. You can actually see, using certain ways, the neurons fire, all of them together, trying to understand how the brain calculates. You can, the entire developmental plan, which cells divide into which cells, in what order. Every cell has a name. We can manipulate them. We can do genetic engineering on them very easily. And for that reason,
Wait,
lots of different
every
amazing things were discovered.
every
Sorry,
cell has a
Abed?
name. Wait,
Every
every
cell has
cell
a
has
name.
a name. So
Every
like
cell has
so
a name.
it's
Yeah,
crazy.
the plan is predetermined. So all the worms are pretty much the same. The nervous system, the brain of the worm, it's connected in the same way every time. So we have a connectome, a map of the entire brain. And it's a powerful model system for studying about lots of different things. And this is why so many Nobel Prizes were awarded for people studying C. elegans. It's the first animal genome that was sequenced before the human genome. Lots of things. What my lab specializes in, and this is most of the work that we've done, about half of the lab works on that, is inheritance, epigenetic inheritance. Epigenetic inheritance means inheritance that doesn't happen because of changes to the DNA sequence, but because of other molecules that get transmitted across generations. And what we study in the lab, and there we are pioneers, and we are one of the leading labs, is that in the worm, when the worm faces all kinds of experimental challenges, it changes the pool of RNA molecules that it transmits to the next generation. So in parallel to inheritance of DNA, in C. elegans, there's also inheritance of small RNAs. These are short sequences of RNA which regulate gene activity. And they obey different rules from the rules of DNA. They show different patterns of inheritance. And the most dramatic thing there is that the DNA sequence is not affected by what happens to you during your life. It doesn't matter what happens to you. It doesn't change the DNA that you transmit to your descendants. But the RNA pool in the worm does change, and this allows the worms to transmit parental responses across generations. Which is the craziest thing you can say to a biologist, that what happens to your life, to you during your life, actually matters to the next generations.
So like we have the DNAs that like they're always like we can predict everything, and like we know what is happening like from like one generation to another, and like we have these like RNAs that like somehow change based on life experience and like events that happening to the worms through life. But we actually know that like also like these RNAs, these like types of molecules are also like transferred to the next generation, right?
Right. So what was discovered in C. elegans is that in parallel to DNA, also there are small RNAs that are being transmitted across generations, and the pool of small RNAs changes in response to what happens to the organism during its life. So, for example, when you starve a worm, it changes the pool of small RNAs, or when you put the worms in high temperature. And because of this change in the small RNAs that are transmitted across generations, the next generations, which are, again, not stressed, living their best lives, are different because of what happens to their parents. And it doesn't last for just one generation. It lasts for multiple generations. And there's a mechanism for amplifying these RNAs over and over and over so they don't dilute. Every worm produces hundreds of babies, and still this faithfully transmits for multiple generations. And it changes our understandings of heredity, and also of what's possible and impossible in evolution. In C. elegans, it's also very interesting, and this is also a contribution from my lab, that processes that occur in the brain of the worm also change the pool of heritable small RNAs. So what the worm think affects the next generation, which is really a provocative thing to say.
And how you measure it? Like how you measure like
so we can the thinking process. So the thinking process that's the beauty, the power of C. elegans. We can manipulate. First of all, we can visualize brain activity. As I said, the worms are transparent. When their neurons fire, you can see it. I mean, using certain techniques. But moreover, you can manipulate the brain activity of the worms. You can activate one particular neuron using light and see how this changes the next generation. As an example, what we did is we manipulated just the two neurons in C. elegans. They have a name. They're called AFD, which tell the worm whether it's hot or not. Right? Normally, the worms like to grow in the temperature, to be in the temperature in which they were cultivated. So, for example, if you grow them in 20 degrees Celsius, and then you move them to 25, they'll crawl towards 20. If you grow them in 25 and then you move them to 20, they'll crawl back to 25. And this behavior depends on the activity of these two neurons. What we did is we manipulated the activity of these neurons using all kinds of ways. And we saw that this is just making them think that it's hot or cold, or changing their perception of temperature, mattered for the next generations. And we can track the path of the RNAs across generations, and also RNA C. elegans moves between different cells. And we can use different mutants that are incapable of transmitting small RNAs across generations, so there it doesn't happen. We have all the controls in the world that we can do, because it's really an animal that you can do anything to very easily, and you can control very well. One of the big advantages of studying C. elegans for studying inheritance is that the generation time is just 3 days. Okay? So you can, in one PhD, you can do lots of generations. If you want to study humans, it's 20 years. That takes forever. And also, everyone has hundreds of babies, or you have a big N. And in addition, the worms, they are hermaphrodites, so they self-reproduce. They make both sperm and egg. So the descendants are almost genetically identical to the mother. So you can separate nature from nurture and genetics from epigenetics. So, yeah.
And how far are we from like fully understand, like, okay, you said both like the DNA, right, and also the RNA and like the system, like how far are we from understanding the different components in C. elegans?
So there are lots of things that we understand in C. elegans, but there are also many missing links, lots of missing links. I was sure you're going to ask, when are we going to see whether this happens in humans? That's probably the question that always comes up. And there the answer is I don't know. It takes time. Also, genetics started from the rules of DNA. Before even they knew that there's DNA, the rules of inheritance were first discovered in peas by the monk Mendel in the 19th century, and then people studied it in flies, and then in bacteria, and so on. It took a long time until we understood that it's DNA, and also that we understood that it's then we understood how it works in humans. In that case, it worked exactly the same in humans as it did in the peas. But when it comes to C. elegans, whether this is a worm-specific thing or it actually changes how we think about inheritance also in humans, we don't know. I'll tell you, however, that 4 out of 5 animals on this planet, if you just count animals, is a worm. Okay, so for modesty, we are the exception. The worms really are everywhere. But also when it comes to DNA, we certainly understand how DNA is inherited, but we don't understand everything that we don't have the entire genome figured out. We know some of it. The part that codes for proteins is a small percentage of the genome. We don't understand, but we can do lots of amazing things with it. We also worked, aside from our work on worms, we also do many other things, some of them also related to DNA. For example, one of the coolest things that we did is we have work on the Dead Sea Scrolls from Israel. I don't know if you're familiar with it, Ravid, but if not, I mean, in any case, I'll tell you about it. Ellen probably doesn't know about it. Probably the audience also don't know about it. So the Dead Sea Scrolls they are the most important archaeological finding ever found in Israel. Okay, and they are these scrolls that were found in the desert in the 40s and 50s, and they were written. So these are scrolls with text. Right? What happens was that there was a Bedouin shepherd in the desert. He had one sheep escape into a cave, and he threw stones at to scare the sheep out of the cave. And he heard the voice of vessels break. He climbed into the cave, which is not easy. I've been there. Okay, it's really a serious thing. And he saw in the cave. Later, they were found also in other caves. Just vessels filled with lots of scroll pieces. They found something like 25,000 pieces of scrolls with text. It's the oldest Bible. The next one is 800 years old. It was written around the year 2000, a little bit before and a little bit after, over the course of a few hundred years. And it's the most important archaeological find that we have, because aside from being the oldest Bible, it is this record from the most important period in the history of Western civilization, where Christianity was formed, where Judaism was formalized. So it's extremely important. People for 70 years have been assembling these pieces of scrolls to try to understand what's written in them. Right, and most of
Wow.
them most of the writing is in Hebrew, so we can actually read it, which is also amazing. Google scanned all of them in 12 different wavelengths, try to uncover all kinds of things, and people did a very good job. What we contribute to this is that we use the fact that the scrolls are written on parchment made from animal skin. I mean, the challenge there is that you have 25,000 pieces. Okay, it's like a puzzle, but it's multiple puzzles. You don't know how many puzzles. You don't know how many pieces. You don't know how many pieces are missing. Some of them are in very good shape. So it's a very hard challenge. What we did is we took advantage of the fact that parchments are written on scrolls that are made from animal skin. And from each piece, we extracted the ancient DNA of the animal from which the parchment was made. We sequenced the DNA of these ancient animals, which is in very bad shape. And then we clustered the pieces together based on the genetic similarity. So we said, this, and this piece, they came from
Oh,
sheep A, and this came from sheep B.
okay.
So they go together, this goes separately, and we did this to sort of assemble the scrolls.
So they
Here,
try to connect like similar like pieces together.
Right. And also piece apart some suggestions that were made in the past, and we made some new discoveries also about the collection as a whole. So we're doing things also with DNA. We're also developing ways to synthesize long DNA, which is a challenge. Sequencing DNA now is easy. Synthesizing long DNA is a big challenge. We are trying to use AI to look, in like LLMs for that were trained on DNA to do all kinds of new things with that. We are working on psychedelics. We are working on evolution, lots of different things. So the lab it's fantastic, the freedom that we have in academia. It's just that we want to make it more fun, more transparent, and more valid as a whole. I mean, the entire scientific corpus should be made more valid.
I'd love to hear all about what you're doing with psychedelics. And I live in Oregon, which is, I think, the most progressive state in terms of having at least decriminalized access to psilocybin mushrooms. And I mean, culturally, the state definitely is full of, hippies, legal weed, decriminalized almost every other drug. I'm just curious what your thoughts are on psychedelics. And I'll also point out that I do believe that Joe Rogan got famous before even podcasting on trying to talk to the world about DMT, the so-called
Mm-hmm.
God molecule, which is considered by many to be the final boss of psychedelics.
Yeah, so I won't tell you too much on that because this is only all stuff that's not published, but and still in the works. We're doing two different things in general. One of them is trying to understand the mechanisms of how different molecules work and the molecular mechanisms. The other is trying to use the worm as a model for studying psychedelics. So, for example, when you treat worms with the LSD, they lay eggs immediately. Okay, this is because it activates the serotonin receptors, which lead to egg laying. Okay, so this is one very very simple essay. It's not too interesting, but you can ask, does this happen also when the worms are mutated and don't have serotonin receptors? Maybe there are alternative mechanisms, okay, that we don't understand. Or you can also look at the brain activity and try to do things that were claimed to happen in other more complicated animals, like look at the entire brain activity of the worm and see whether it is more synchronized and how it changes and things like that. So this is the type of thing that we try to do with the worm. And I think it's just a fascinating thing. Personally, I never tried any psychedelic. I cannot say anything about the experience itself. I'm afraid to do it. But it's uh, but we know from the cultural influence that this is an amazing way to probe brain activity and also higher dimensions of brain activity. And there's a total disconnect between this, the reported experiences, and the understanding of the molecular and neuronal mechanisms, which are still very, very preliminary. So I think there's a great opportunity. Of course, everyone is jumping on that, but I don't think so many people are jumping on that using very simple model organisms that are actually trackable and you can understand.
I want to talk a bit about like how to call it auto research. Like you said before that like there are a lot of like experiments that like we still need to do and like we don't understand even in such a simple model as the elegans. Um and my question is, so like is it like all the experiment that you should do, like, and just a matter of time and resources and people to do it, or like there are some like open question that like we don't know yet, and like we need to come up with like new paradigm. um and then like we talk about how like it's related to auto research, because like this is like exactly the question I think that like we are asking, like if we can actually like make auto research like using models and like these like self improvement models and all these topics. So yeah.
Right. So first of all, I mean, I think, and this is, it's aligned with our entire philosophy. Also, what I do in the lab, but what I do also in QED, I think that scientists want to pursue their own ideas. They don't necessarily want AI to give them the ideas of what to go and study. And I think that this is a very good thing. Right? So there are lots of AI companies and also researchers saying, AI will create the hypothesis and will pursue them. I think that I'm sure AI will also come up with great ideas. Absolutely. There's no reason it won't. But I think that we want to preserve this human element. If we're thinking about what about research do we want to preserve? I think we want to preserve this human curiosity, which is in many cases totally not practical and not and just hard to so for example, I mean, you go and you study black holes, and because of that you go and you invent a microwave. I don't know, because you started by something which is totally just for human curiosity, and it ends up being practical. We never know. I think that if we let AI just go and find the most useful things, probably won't go to some avenues, and we'll narrow the search. So and I think also that's the whole pleasure of being scientists. And people will not want to be scientists if they will be just pipetting for the robots, or just doing the non-interesting things. So I think that it's very strategic. Although in QED we want to preserve this and have people pursue their own ideas, but just in a better way. We want to sort of, instead of replacing them with an AI scientist, we want to build an Ironman suit for the researchers to do better. About whether there are more questions to ask that we don't even know, or directions that we need to follow that we never thought of. Absolutely, I'm sure there are. I wish I knew which ones they are. But as I said, biology is just limitless. We have no idea. Let's go back to the model organism. I said that lots of the success of biologists came from studying just a handful of model organisms. C. elegans is one of them, certainly not the only one. There's the fly, E. coli bacteria, Arabidopsis plants, zebrafish. Just a few models that everyone study, and you get the most bang for your buck by choosing a model organism and just focusing on and drilling and just exposing the secrets that it offers. We had no idea that these exist, but the amount of animals and organisms you have on this planet, each revealing the things you never thought about, and emphasizing a particular part of biology that was just perhaps very minimal in some other organisms like humans, but very robust. Like in C. elegans, the mechanisms for transmitting RNAs are there. And they are super elaborated. They are dedicated machines for doing that. They are in front of our eyes, and we can characterize them. It's possible that the same thing happens in humans. We don't know, but perhaps it's much more subtle or less important, and it's very hard to understand. In C. elegans, it's like a land allowing us to explore it. So I think just by moving to, this is just the most trivial thing, just by expanding the repertoire of animals that we study, or organisms that we study, we'll find new questions, totally new questions. And there's this illusion, and this is part of the hype that I mentioned, of some companies that and you can just have AI just look at the data that is out there, perhaps also automatically just collect data, and it will connect the dots, and everything will be figured out. And I think it's really not the case. Because there are so many details missing. It's really an ancient myth thing. I don't know if that's a word. That's a word, myth thing. I don't know. It's funny if it's not a word, but it's a misconception. It's an ancient misconception that even I think Galileo talked about. When Galileo talked about, I think it's a book of nature or something like that. We talked about the fact that people interpret reality in his time all based on Aristotle's writing. Say the scientists of his time thought that everything is there in Aristotle. He wrote about, and they can just look at what he wrote and connect the dots. Mm-hmm. And he compared it to these paintings of Arcimboldo. This painter who used to paint like a human face from fruits or different vegetables, right?
Yeah.
So
Yeah.
these are the elements, and you'll force them to build a face. So this is I think this is very similar to trying to understand. We'll figure out nature, but what we have now, we just try to connect it, put it in place, and we'll figure out all of the mysteries of Unity. It's just the details are missing. You need a deep understanding of the processes and the logic, and to create a very complicated models of how things actually work. And and the most interesting things are just not there in the data. I mean, all we can do is we can point you in the right direction to where there's a gap, and then you go and you do the experiment and you look for it. And so absolutely, I think we're not in any way close. It doesn't mean that we won't be able to do useful things, but we are very far from solving everything.
So, so a few weeks ago, like we talked with Max Welling. He has now like a new company about like experimental like wet labs, that like how to close the loop and like to make like to suggest experiments and then like to run it automatically. Um, so what do you think like is the more I don't know, more interesting direction? Do you think like it's in the automation side of AI, like automation of like the experiments and like to make them faster and reliable and cheaper, or like for like the review side as you suggested?
So I think, I mean, it's a whole ecosystem, and we need all of that. There are parts that needs to be automated. Biology is also very tedious and laborious, and you do endless experiments that are hard and would also gain from automation for sure. And AI can help you with the calibration of that, and also explore lots of different options. Absolutely. But in addition to the creative part, and you need AI. So it's not only about review, it's for finding what's valid, what's invalid. I think we need to build this validation machine to understand what we can build on. Say stand on the shoulders of giants. We need that. In biology, it's just it's extremely the corpus is really noisy, and it's very hard to build on them. And it's a science that requires building on others all the time. And if the foundations, if people are talking about biology being programmable in the future, if you want to program it, you need to have the building blocks solid, and you need to understand what are the building blocks. Otherwise, it will never be programmable. But for it to be an engineering problem, we need some solid ground. So some people need to work on automation and calibration. Some people and about better ways also to find patterns in existing data, and some people like us in QED need to create this validation machine that will just tell you what you can trust, and and sort of reorganize the entire scientific corpus so it will be reliable and will allow you to progress. We need all of that.
So I have a question, and maybe, I like to try to ask the very hard questions in a field. And I think that what's considered among the biggest unsolved problems in all of biology is this question about what is the source of origin of life, I guess, or I believe they call it abiogenesis. Do you have any opinions on what the actual origin of life might be?
No, so I'm certainly not an expert. Okay, I can tell you from my very little understanding. From just people talked, and of course, it's a very old concept already about an RNA world. So it's clear that RNA was there before DNA, and and RNA as a molecule that helped the origin of life is appealing also because RNA, unlike DNA, it's not only information, it's also a molecule that acts as a molecular machine that can do things. DNA doesn't do anything, it's just the information. Okay? RNA is both information, and in some viruses, for example, there's only RNA and not DNA. Okay, and so it's the only molecule of information, but it's also a machine. There are RNAs that works in catalysis and so on. So RNA was certainly important in the early in life, in the origins of life, but it doesn't mean that it is certain that is the first molecule that started it all. So there could be short peptides and so on. And since I'm not an expert, I don't want to talk nonsense, too much nonsense.
So I have like one final question, like before we end. So like, we talked a lot about like review and how to make like review with AI, but there is the problem at the end that like even if you say that like yeah this is like high quality in review, there is the problem of like I want or like sometime or some people wants to get a review from real humans. Like we see it so at least in AI, we see it like in conferences that like that these days basically like you have two tracks, like one that you can get review from that help AI help to generate, and one that you can't, and like other there are some like conferences that if they detect some hallucination in the reference, for example, like they reject your paper. Even though, right, it can be like there were mistakes and errors even before AI, but like now they say, okay, if it's a reference, you made up a reference, like you change some names in the reference, it's probably like you generate the paper with AI, and now we can't trust your paper anymore, right? So what do you think? How you see this
Right.
different
So
point of
you asked a
views?
few different things. A few different things. First of all, I think that I'm all for humans being involved in review, and I think that there might be certain things that humans will always be better at than AI. I'm not sure, but perhaps. Okay. And absolutely involve them. I mean, the two ways should be complementary. So great. Okay. And when it comes to fear of AI being involved in review, you need to separate two different tiers. One of them is that the quality of the review will be bad. Okay. The other is that some element, some human element will be lost from the equation. Then we need to understand what is this human element we want to preserve. It's not an easy question. Okay? When it comes to review. But the might I think that and then you commented about the fact that people are currently, and this was visible in many scandals, disqualify papers that were written by AI or grants that were written by AI, or disqualify abstracts that were submitted to conferences because AI has been involved, or also are mad at reviews that were generated by AI. Okay? The question is, why? Okay, if the review or the paper were slop or just poorly done, okay, then sure, it's just low quality. What is it about the fact that it's AI that is so infuriating to so many people? And
Yeah.
and the thing that is infuriating is that people don't take responsibility and don't go over the product that they actually submit before submitting it. I think it's a little bit like if so. So let's say you involve AI in the writing of your paper, okay? And it's messed up. It hallucinated the reference or just made something horrible. I mean, in the end, it's your name on the paper, so you should be responsible for what you're submitting.
Yeah.
If you go, I don't know what is the theoretical problem with the fact that you submit a paper that was fully written by AI that you just watched over, and then you checked and it's valid and it made some discovery. There might be some problem there of then getting who gets the credit, but that's a different question. Okay?
I
And I
agree.
think it's the same. It's a little bit like when you have students in your lab. If a student cheats or hallucinates or messes up in any way, the PI is responsible. It's true that although the student will be in a big problem, but also the PI will be held responsible. And then people say, okay, but the AI is generating so much so fast that it's just impossible to actually review, and that is a problem. So you need to the quantities needs to be that you publish and you put out there in the world needs to be quantities that you are actually able to monitor. Perhaps some of the solution, because we don't want to limit the quantity if it's good, right? Some of the
Yeah.
solution is also to automate some of this monitoring.
So I must say that like I have mixed feelings here because like from one hand, like if your student is making mistake, right? Like some put like your own reference or something like that. So before AI, like it was yeah okay, make a mistake, but if it doesn't like change something fundamental in the paper, so okay, fix it, right? Um, but with AI, like now everything is hallucination. So like we know it can't be that like there is some hallucinations in the paper, everything is wrong. Um, but from other hand, I kind of like get it, because I think the main problem with AI is that now, like, we don't actually know, we don't have a good understanding about its mistakes, right? Because like now, like we know that the student like will probably like doesn't put something like I say it like intentionally, right? Like he doesn't put like wrong results in intention, and like this is our
We hope.
assumption. It may
We
be
hope.
true or
Yeah.
not.
Yeah.
We hope, yes. But like we kind of like have this like common belief that people are good, and like they are not faking like totally the results, right? Because like I'm sure that like if you like work really hard, you can fake results, right? Like you can
No,
make
no,
like
I totally agree that I think that, I mean, also when we're talking about the reasons that the scientific corpus is noisy in there, that there are many things you cannot rely. Some of it is because of fraud, absolutely, but I want to believe it's a small percentage.
yeah,
The vast,
yeah,
the bigger problem is just science is hard. That is the
yeah.
big problem. Yeah.
But in AI, no. But the thing with AI and like with model is that like we don't have this like this belief, right? Because like the model actually like it will insert like the wrong reference, not like by mistake, or like because he wants to prove something, or like he will change the results because he wants to make them better. Like he doesn't have this good faith that we believe for humans.
Right, and I mean, I think it's a little bit like the same problem is there with self-driving cars. Now,
Mhm.
there'll be very few disasters, but even one is enough for paralyzing the entire industry. And so we take these mistakes that that the machines do differently than we do when it comes to humans, and part of it is about, like you said, exactly understanding the source of or the intention behind the mistake. But I think the gold standard would be it's always the human's responsibility. If it's their name on the product, paper, ground, whatever, it's their responsibility. And if they are producing more text than they can, uh, and more claims that they can actually um, put their names on, and guarantee that it's a problem.
Yes, I agree.
Do you
Okay, I think like, oh, Alan, you want to?
do you think humans will retain a role in AI research in 5 or 10 years?
In AI research. So there, I'm really not an
Or in
authority.
biology?
In biology, yeah, absolutely, absolutely, absolutely. I mean, probably we'll need to also reinvent ourselves, and we'll need to change. In some ways, I think it would be good. I'm actually optimistic. I think that the last thing as a biologist I'm afraid of is being replaced by AI. I think it just creates more opportunities for me.
This is because you have tenure.
Also, yes. Not only, not
Yeah.
only. Yeah.
Okay. Oded, do you have anything to add before we finish? To advertise
Uh, and no, I
besides your
mean, I think
company?
no, no. I mean, I don't have anything to advertise beside QED. I'll say as a sort of a disclaimer that currently it works for life sciences. Life sciences. Okay, so I know lots of people who listen are not biologists, but for biology, I think it's very good. And if you are a biologist or you have a biologist friends, use it. And we are sort of trying to now switch to developing in the open and just add lots of different things every week. So that is all I want to say.
Okay, great. Thank you so much for coming. It was a really pleasure for us. And
So thanks a
thank you
lot
for the
for
audience.
hosting me.
Thank
Thank
you.
you very
And
much.
bye bye.
Thank you. Bye bye.